speakers. We'll go ahead and start. Our first speaker is Elsie Platzer from UC Davis. So with that, go ahead and get her presentation up.
Hi, everybody. Welcome to my talk today, Silverside Gold Mine, Non-Native Fish Use of Suisun Marsh Tidal Restorations. My name is Elsie Platzer. I'm a second-year master's student at UC Davis, and I study wetland restoration in Suisun Marsh. Suisun Marsh is a really intriguing ecosystem. For one, it's the largest contiguous tidal marsh on the West Coast, but because of a series of human interventions, including drainage for agriculture and damming and hydraulic mining and many non-native species introductions, it really no longer resembles the Suisun Marsh of the past. It's gone from a network of seasonal wetlands and floodplains to a very channelized system, a novel ecosystem. And because of this, there's a lot of tidal restoration projects being funded to sort of reverse the habitat losses and improve ecosystem service provisions in the region. So my personal research goals are threefold. First of all, in collaboration with other projects like the Suisun Marsh Fish Study, I'm hoping to characterize fish assemblages across all different Suisun Marsh habitat types, and more on those habitat types in just a minute. Secondly, I want to work to identify mechanistic relationships and predictors of fish habitat use. So those might be biotic variables like primary and secondary productivity, or abiotic variables like water quality. And finally, I want to use these data to sort of inform restoration practice throughout the marsh. So when I'm talking about habitat types, I have three distinct types in mind. The first is tidal marsh or restored marshes. These are characterized by heterogeneous landscape features. So they're a mixture of small channels and marsh plains with emergent vegetation, and they have open tidal access. They are tidally connected. The second habitat type is the managed wetlands, also called ponds. We call them ponds. So these are diked wetlands, basically. There are gates that managers use to control inflows and outflows, and they follow a seasonal flood drain cycle. So in the fall, they'll flood up, and then there'll be a hunt season for ducks because they're usually managed for recreational duck hunting. And then as it starts to get towards summer, they'll be drained. So because of this, they have longer residence times between circulation, and that leads to increased primary productivity. And then finally, the last habitat type is slough channels. So these are our historic habitat, usually wide, deep channels with not a lot of edge habitat. And these are really often tidally flushed, so daily tides coming in and out. So for my project, I'm using a combination of methods. I am trying to get a sense of nearshore fish assemblage through beach seining, and then I'm supplementing the seining and the fish sampling with zooplankton counts and water quality surveys. So looking at temperature, turbidity, salinity, chlorophyll, and all of those things. And together, I hope this will give me a more holistic understanding of the biotic and abiotic conditions of the different sites I'm looking at in the marsh, and how that correlates to where the fish are. So my project is lovingly titled WTF, which stands for Where's the Fish? And here's a map of our sampling locations. So we have 4 distinct spatial complexes. Basically, we're just trying to be at all 4 corners of the marsh to cover the variations in like landscape features and typical salinities that occur across Suisun. And I'm still in the process of sampling, and I will be for a while, but I thought I'd share some of my primary findings with y'all. So this simple graph is of fish catch per unit effort since we began seining in November 2020. And you'll see there's already a dramatic trend that's apparent. So 2 fishes are very dominant in the restoration and sloughs. We have Mississippi silversides that are dominant, and then in the managed wetlands, 3 spine stickleback are dominant. And this trend was also apparent in a study that my colleague Brian Williamson did in 2021, comparing fish assemblages in Blacklock Marsh, which is a restored wetland, and Luco Pond, which is a managed pond. As you can see here, silversides are in Blacklock, sticklebacks largely in Luco Pond. So the fact that this trend, which was observed by Brian, is continuing to be apparent throughout the whole marsh scale is pretty telling. So in order to kind of delve into why this might be the case, we should take a closer look at the life history of these 2 fishes, starting with 3-spine stickleback. These are a native resident fish. They're invertivores, so they're going to really enjoy the managed wetlands where secondary productivity is high and there's lots of amphipods to eat. And also the managed wetlands provide protection from predation because those gates that are used to restrict the tides also restrict access from larger predatory fish like striped bass, which can't come in as easily. So they have a pretty good life in the managed wetlands. Mississippi silverside, on the other hand, these are non-native fishes and they kind of get a bad rap because they've been known to predate on delta smelts, eggs and larvae. So somewhat similar to the sticklebacks, they're small, they are littoral fish, they like warm water and edge habitats. So you would think that they would kind of also do well in this shallow managed pond, but they don't. And the reason for that has to do with the interaction between hydro period and reproductive biology for these silversides. So here's a graph of total silverside and stickleback catch per unit effort broken down by month. And you can kind of see how the numbers of silversides increase over the course of the year. That's because their spawning period is between May and October. So this is a critical time for them to maintain and build their population. But interestingly enough, the pond drainings happen all by the beginning of August. So at that point, all the managed wetland sites are dry. So when silversides are spawning, they can't go to the ponds because the ponds are not there, they're drawn down. So this kind of forces them to move more into the restoration and slough sites. Whereas sticklebacks spawn a little bit earlier, mid-March through July. So they're able to utilize the managed wetland ponds. Also interesting to note is that the stickleback population drops off really intensely by the beginning of August. And that's because as the ponds are drained, the sticklebacks come flushed out of these outfalls, and the predatory fish know to sit and wait for all of the flows to come out so they can basically scoop up the stickleback. So a lot of them die due to predation at that point in time. So basically what I'm finding with all of these preliminary data is that the tidal restorations are catering a lot to non-native fishes like silversides. And if restorations are turning out to be a Silverside gold mine, then what should the response be from the scientific community, the management, and conservation communities? Well, like it or not, silversides are here to stay. They're pretty much the dominant fish in Suisun Marsh near shore. So it's time to get really creative about solutions that move beyond like a breach and leave approach, as we call it, where you just open the levee and restore tidal connectivity and hope that the ecosystem rights itself. In the context of a novel ecosystem like Suisun Marsh, it doesn't always pan out that way. So instead, we can maybe start to explore habitat benefits in managed wetland ponds, or think about design modifications for already restored areas, or some other creative solution. And I want to add one more caveat to this, which is that we shouldn't use a simple conceptualization of the ecosystem where native fishes are good and non-native fishes are bad. And recently I read a really wonderful article by Julie Stromberg called In Defense of the Immigrant. And Julie Stromberg is a plant ecologist who has worked a lot in arid riparian systems to manage the invasive plant tamarisk. And the thesis of this essay was basically that the invasive species are a symptom of the larger ecological problem, which is that the invader is us. So explicitly calling out systems of settler colonialism and dispossession and resource extraction as the root cause, and sort of calling for a new approach to invasive management where we are acknowledging these larger scale ecosystem changes that have occurred, and B, we're not demonizing the non-native species that just happen to thrive in the highly altered habitat that we created. So maybe with this in mind, we can find a silver lining to all of the Silversides that we're finding. So at least in coastal California and with other fishes in the Silverside family, we know that certain birds, protected bird species, really enjoy eating Silversides. So we're starting this pilot project at Montezuma Wetlands, one of our tidal restoration sites, that'll look at eDNA in tern feces and see if there's evidence that the terns in Suisun Marsh are using the Silversides as prey. And that would be really cool because it would point to the integration between native and non-native species, a native protected bird like the California least tern and a non-native nuisance fish like the Silverside into a single novel food web. And that would be pretty exciting. So we hope to see some fun results from that. So that's all I have for you today. Thanks for coming, and please let me know if you have any questions.
Okay. Thank you, Elsie. Wonderful presentation. I really enjoyed that. Feel free to put questions either in the Q&A, or feel free to raise your hand if you have any questions. We've got about 7 minutes. I'm not seeing anything. So Elsie, I have a question for you. With that project, where was it plovers that would potentially eat the Silversides? Would there be a concern that they would also eat Delta smelt then, or other fish like that?
Well, not particularly. I think I'm not a bird expert in any way, but I think that a lot of the piscivorous birds that are in Suisun Marsh are just interested in what has the most biomass, what they can get really easily. And Silversides are everywhere. They're super abundant. And they're also kind of the right snacking size, which sounds weird to say. And they're not, they don't have any spines like some other fish do. So they might be preferable, but this is all very hypothetical at this stage.
Yeah, no kidding. So what's the timeline for starting that project, or have they already started?
We're just very, very early in talking it out, hashing out a lot of the details. People who are working with us from Andrea Schreier's lab, also at UC Davis, have secured a few grants to just do a bunch of the very basic eDNA work to kind of test if you can pull apart fish DNA from bird feces, or if the bird DNA will overwhelm it. And so just very early, trying to figure out what the methodology would look like.
Okay. Excellent. So I see we have a question. It's from Melissa Riley. Elsie, do you see signatures in your fish catch of Silversides and stickleback that correspond to the drain leach cycles in the late winter / spring that occur after waterfowl season versus full drawdown in the summer? I can repeat that if you need me to.
No, I think I can answer that. So usually when the ponds flood, it takes a few months for the fish to arrive. They don't immediately colonize the way that in tidal restorations, fish seem to arrive very, very quickly. But in the ponds, it takes like 2 to 3 months for them to start showing up. So when they flood up in the fall, for the first few months, we'll catch no fish, no fish, no fish. And then we'll start to get sticklebacks. And then, especially this past year, the ponds have been draining really early because of the drought. So as they drain down, our fish catch goes up because there's just more fish in smaller like pockets of water. So they're clustered as the total area of the pond is decreasing. And then right before drain down, there might be a decline. So that's pretty much the cycle of it that we're observing.
Okay. There's more questions coming in. I'm gonna pick this one, Timothy Malinich. If eDNA doesn't work, can you use fatty acids or stable isotope methods to investigate what may be eating silversides?
I have no idea. That is something to ask our collaborators, but I'm not
no expert problem
in that.
So here's one from Matt Young. He says, you may have addressed this, but I arrived late. To what extent do you think these communities represent a stable endpoint versus transitional communities that may evolve with the restoration project?
Yeah, that's a super interesting question. And all of the restoration sites that I am sampling at are very, very young restorations. They're all, I think, breached within the last year or year and a half or so. So there's still an opportunity for dynamics to shift, and a lot of new things are going on. But at the same time, silversides are not decreasing, and there's no force that would really cause them to see a population crash. So I don't know what would shift that aspect of their dominance. But I do think there's space for the communities to change in the restorations as maybe more emergent vegetation establishes, or as hydrodynamics change with new channels being cut or new ponds forming and things like that. So nothing is set in stone, but I'm unsure of how much it's transitional.
Right. Okay. We have about 1 minute left. I'm going to go ahead and ask this last question, but we may have to cut the answer short. Has anyone looked at whether Mississippi silversides could be a high quality food for salmonids compared with other forage fish like northern anchovy? And feel free to say that's out of your wheelhouse too.
Yeah, I'm not sure. That would be pretty interesting to look into.
All right. Thank you so much, Elsie. Very interesting topic.
Thanks.
I think at this point, we'll go ahead and queue up the next talk. That's Melanie LaCava, also from UC Davis. So go right ahead.
Hi, my name is Melanie, and I'm excited to speak with you today about reproductive timing in delta smelt as it relates to a captive spawning program. As many of you know, delta smelt have been in decline in the San Francisco Estuary for several decades. In 2006, the UC Davis Fish Conservation and Culture Lab, or the FCCL, started a captive spawning program to maintain a refuge population that would serve as a safeguard against extinction and also provide a source population for future releases of cultured fish into the wild. One of the primary goals of the spawning program is to maintain phenotypic and genetic similarity of captive fish to their wild counterparts, so that when fish are released into the wild, they can easily integrate back into the population. The lab I work in, the UC Davis Genomic Variation Lab, or GVL, works with the FCCL to genetically manage the spawning program to strive for this goal. But any time that you bring animals into captivity, there's a risk of them becoming genetically adapted to captive conditions in a way that can be detrimental once they're released back into the wild. So we wanted to explore how a few traits related to reproductive timing in delta smelts have been affected by more than a decade of captive spawning. I wanted to start with some background on why we care about genetic adaptation to captivity, using research on steelhead as an example. This study by Christie and colleagues evaluated the reproductive success of steelhead born to wild parents, or those with 0 generations in captivity, versus parents in captivity for 1 generation. And you can see that captive parents had higher reproductive success, demonstrating adaptation to captivity in just 1 generation. Now, they took this study a step further by evaluating the reproductive success of parents in the hatchery compared to the reproductive success of their offspring in the wild. And here we can see that parents with high success in the hatchery had offspring with low success in the wild, and vice versa. And so together, these results demonstrate rapid adaptation to captivity that can lead to lower fitness in the wild. So if we want fish to succeed in the wild, we need to do everything we can to limit adaptation to captivity. So now I'll walk through how the delta smelt spawning program works so we can think about how adaptation to captivity might occur. Every spring during the spawning season, the FCCL collects fin clips from sexually mature fish, and then the GVL can genetically identify the parents of that tagged fish. And this way we have a complete pedigree of all the fish in the program. Then the FCCL checks for females that are ripe, meaning that they're expressing eggs and they're ready to spawn. And the GVL tries to identify tagged males that are good genetic matches for the females. We aim to avoid fish that are closely related, and we aim to have relatively equal representation among fish families in the spawning program. Every year, around 250 pairs of fish are spawned over the course of several months. And while we do our best to choose genetic matches, we have some logistical constraints because in any given week during the season, we can only choose from females that are ripe and males that are tagged. And therefore, the amount of time it takes fish to become sexually mature could influence whether or not they are included in the spawning program, which is why we were particularly interested in studying reproductive timing. In addition to these logistical constraints, we can also predict how changes to reproductive timing in captivity could affect delta smelt once they're back in the wild. And one risk is truncation of the spawning season. The FCCL spawns delta smelt during the peak months of the season, but fish that are mature outside of these few months are unlikely to be part of our spawning program. So we could potentially miss out on fish that mature very quickly or very slowly. And we don't know how important these outlier phenotypes are in the wild. Another risk is some kind of mismatch with environmental conditions. We know that environmental conditions are highly unpredictable in the wild, so having some diversity in when delta smelt are ready to spawn seems most beneficial to the long-term success of the population, which is why it's worth investigating how captive spawning has altered reproductive timing in delta smelt. For this study, we are representing reproductive timing with a variable that we're calling time to maturation, which we defined as the number of weeks it takes after a fish egg is fertilized for that fish to develop into a sexually mature adult that is expressing gametes. The first thing we wanted to evaluate was how variable the time to maturation is in captive delta smelt. If we look at this histogram of time to maturation, we can see that most fish become mature roughly 1 year after they're born. This red line represents 1 year, or 52 weeks, and this makes sense, right? The spawning season occurs every spring, so fish need to be mature around 1 year of age. But we can also see that some fish are maturing as quickly as 9 months, up to about 16 months. So there's a lot of variation in this trait to explore. Now that we know that this variation exists, we can ask how the time to maturation has changed over the past 10 years. One prediction is that captive spawning might favor faster maturation. If this curve represents the range of time to maturation a population starts with, this prediction would shift the whole distribution to be faster. And we were interested in testing this prediction because of those logistical constraints that I mentioned. Specifically, early in the season, there's a limited number of ripe females, so most of them are chosen to spawn. But as it gets later in the season and some fish families become well represented, females that become ripe might not be selected for our program. And so there's a potential that we're favoring fish that are ready to spawn early in the season, and so we could be pushing the population to mature faster over time. We evaluated this prediction by looking at the time to maturation across the past 10 years to see if it became faster over time. However, when I show you the data, we don't see any significant decline in the time to maturation. In fact, it looks like time to maturation really hasn't changed much in the past 10 years. So we don't see support for faster maturation over time, but we also wanted to explore a second prediction that captive spawning could be removing outlier phenotypes, meaning fish that mature very fast or very slow, because we only spawn fish during the peak months of the season. Here you can see we'd predict the distribution of maturation timing to get narrower if we were eliminating outlier phenotypes. Here we're looking at the standard deviation of time to maturation to see if there's an overall decline in the variation over the past 10 years. And our data do show some decline in standard deviation, though I'll note that it's not statistically significant. So here we're seeing some support for that prediction that captive spawning is reducing variation in how long it takes fish to mature. In terms of relating this change over time back to potential genetic adaptation to captivity, we wanted to explore how heritable this trait is in delta smelt. To measure heritability, we need to think about what determines how long it takes each fish to mature. A phenotype, like time to maturation, is determined by some combination of the organism's genotype, or what's encoded in its DNA, and the environment. And traits vary considerably in how important the genotype versus the environment is in determining the observed phenotype. Just as an example, in humans, eye color is highly heritable because it's largely determined by genotype, and there's much less influence of the environment. So we're interested in this genotype part of the equation to figure out how heritable time to maturation is in delta smelt. And we can estimate heritability by comparing how long it takes fish that are related to each other to mature, using the complete pedigree we have for fish at the FCCL. In this plot, we're going to see what proportion of variation in the trait is heritable in each of the past 10 years. And just for reference, a value of 1 would indicate the trait is completely heritable, while a value of 0 would indicate it's entirely determined by the environment. When we look at our data, we see an overall decline in heritability of the trait over time. In early years, we see heritability as high as 0.7, indicating that 70% of the variation in time to maturation is heritable, and that's quite high. In more recent years, we're seeing only about 10% of the variation in time to maturation is heritable. Now, this decline in heritability over time might be related to the loss of variation in the trait that I showed you on a previous slide. Maybe by losing some outlier phenotypes, we've lost some of the heritable variation in the trait. If we come back to our equation here, as the genotype becomes less important over time, then the environment must play a bigger role in determining the time to maturation. Now, measuring the influence of the environment is challenging because there are so many different factors to consider. But one aspect of environment that we can assess is what we call maternal effects. Maternal effects account for variability in the observed trait that's influenced by something coming from mom that is not heritable. For example, say we have one mom on the left that's in great body condition, and so maybe the eggs she produces are big and healthy compared to another mom that's in poorer condition and therefore produces lower quality eggs. Now, this difference in egg quality is not genetic. Rather, it's related to the body condition of the moms. And so this is an example of a maternal effect that could influence the development of her offspring. If we come back to our heritability plot, we can now look at how much of the variation in maturation timing can be attributed to maternal effects in gray. And interestingly, we see an overall increase in the importance of maternal effects over time. In recent years, we're seeing maternal effects account for up to 40% of the variation in time to maturation, which tells us that something about the female fish being spawned is having a big impact on how long it takes her offspring to mature. In the next stage of this project, we're planning to investigate how the condition of the mother affects how long it takes her offspring to mature, using morphological data collected at the time of spawning. If we come back to these two hypothetical fish, we might expect the difference in body condition of these fish to tell us something about the quality of their eggs, and this might relate to how quickly the offspring develop. So, for example, one of our predictions is that maybe offspring from moms with better body condition are able to mature faster than offspring from moms in poorer condition. So, in summary, we found some evidence of a loss of variation in time to maturation, and we found that heritability of the trait declined over time, while maternal effects increased over time. And we're excited to continue exploring these maternal effects moving forward. But what do these results really mean for delta smelt? Despite some evidence of loss of variation, we're not seeing any drastic changes to reproductive timing, even after 10 years of captive spawning. And this demonstrates the overall success of the careful management of the population. And as we discussed, we care about adaptation to captivity because it can affect fitness in the wild. And since we don't see any drastic changes to maturation timing in captivity, we don't expect any impact on fitness in the wild related to this trait, which is great to have just one less thing to worry about for the success of these supplemented fish. And with that, I would like to acknowledge many organizations and individuals that contributed to this long-term data set or provided funding for the program. And thank you for listening to my talk.
All right, thank you so much, Melanie. With that, we'll take questions. Who's got questions for Melanie? Either raise your hand or you can add those to the Q&A at the bottom of your screen. Okay, from Mandy Finger. Great talk. I wonder if when you figure out traits that are related to maturation, it can be used to predict where delta smelt will be / when they will spawn in the wild. You want to talk about that at all?
Let me read this quickly.
Do you want me to repeat that?
I can see the question.
Okay.
I'm trying to figure out what she means. I guess I'm not sure exactly what Mandy. Oh, good. She acknowledges a hard question. I guess one thing is if we know something about how long it takes fish to mature, we might be able to incorporate that into these releases that are happening, right? When we're thinking about what time of year to supplement the wild populations, maybe this is one more factor that can be considered among the very extensive list that I know is already being considered. But obviously, by releasing fish this winter, as we heard about this morning, the goal was to release adults that would reproduce this spring. So knowing about reproductive timing can definitely influence the success of these fish in the wild, which if we're releasing them, the goal is to have them reproduce and continue on. So we can think about that more.
Yeah, and Mandy followed up with, I'd just like to see the people that are looking at where/when smelt spawn, look at the data and research we are conducting in the hatchery. So basically, I think what she's getting at is like comparing EDSM catch data or other programs to what you found.
Mm-hmm. Yeah, it would be great to have any information from the wild about when fish are being caught. I know they're recording things like body condition and morphology, but also if they're expressing eggs or milt. And so if there's any data to be able to compare what's going on in the hatchery with the wild, that would be really interesting.
And then it looked like Evan Carson had his hand up. Evan, can you go ahead and unmute and ask your question? Maybe not.
Okay, there we go. Yeah, so there are two interesting things here with regard to spawning in the wild. One is if we know something about the timing of maturation and when those fish were produced, for example, when they're released, that may give fish some advantage of finding where they need to be, when they need to be there to spawn. So as they're exploring the delta, we know they move around now from EDSM catches. And so just having more information about that would certainly be worth considering in how we release fish. And also as something of a twist on the maternal effects in the hatchery is we're facing the possibility that because fish do go out healthier than what we've seen of wild fish, that there may be some advantage to fish being in better condition such that larvae stand a better chance not just of hatching but of surviving. So there are potential advantages as well as disadvantages and how that shakes out. We're in the infancy of this, but there's really a lot of potential with this type of study and a lot more work to be done.
Yeah, I think what Evan just pointed out is important, right? Is that if heritability is declining over time and there's more maternal effects, if we're able to produce big, healthy moms that are being released into the wild, then maybe their offspring will have more success once they're out there, which is positive.
And then I think we have time for just one more really quick question and answer. Tyler Pilger wrote, is there going to be an effort to look at genomic basis for maturation timing? And feel free to say if that's out of your wheelhouse too.
Yeah, I think that's a great question. For this project, we haven't explored that yet. We have a ton of different genetic projects going on for Delta Smelt in our lab, including some really interesting work on domestication selection by Enzi Habibi, who gave a lightning talk a couple of days ago, where she's looking at the whole genome and looking for signs of selection. And so at some point, maybe our projects will kind of merge together. But at this point, we're really trying to take advantage of the pedigree that we have for the hatchery, as well as all this phenotype data related to when fish are spawned and how long it takes them to become mature, and just kind of see where it takes us.
Okay, thank you, Melanie. That puts us right at 3:30. Thank you for sharing all that information. We'll
Thanks.
go ahead and load up Ryan McKenzie from U.S. Fish and Wildlife Service as the next slot.
Hello, everyone. Thank you for attending my talk today. My name is Ryan McKenzie, and I work for the U.S. Fish and Wildlife Service in Lodi, California. And today I will be discussing the preliminary results of my study assessing biotic homogenization of the Sacramento-San Joaquin River Delta. First, I would like to make a few acknowledgements. I would like to acknowledge the funders of this project, the California Department of Water Resources and the U.S. Bureau of Reclamation. I'd also like to acknowledge my collaborator, Brian Maharja, and the individuals of the U.S. Fish and Wildlife Service Delta Juvenile Fish Monitoring Program for collecting the data used in this study. I would also like to thank the Interagency Ecological Program for putting on the IEP workshop and allowing me to give this presentation today. So if you're like me, you're probably wondering, so what exactly is biotic homogenization? So biotic homogenization is the process by which two or more spatially distributed ecological communities become increasingly similar over time. Here we will give an example of biotic homogenization. Take Community 1 and Community 2. At this point in time, Community 1 and Community 2 have very different species compositions. But over time, and through the process of biotic homogenization driven by species extinctions, migration, and changes in species assemblage, what we will see is that the species composition between Community 1 and Community 2 will become increasingly similar through time. There have been a number of causes of biotic homogenization that have been documented in ecosystems around the globe. Many of these same causes have also been documented in the Sacramento-San Joaquin Delta, including species invasions, species declines and extirpations, and shifts in species assemblages. So in this study, we set out two main objectives. Our first objective was to assess regional biotic homogenization of the Delta's nearshore fish community over time. And our second objective was to determine which fish species correlated to the regional beta diversity over time. In order to assess biotic homogenization, we utilized the U.S. Fish and Wildlife Service Delta Juvenile Fish Monitoring Program Beach Seine dataset. The Beach Seine survey has been conducted year-round since 1995. It uses fixed seine sites sampled weekly or bi-weekly throughout the San Francisco Estuary. And currently, this data is available for download on the Environmental Data Initiative Data Portal website and can be found by searching DJFNP in the search engine. Before analyzing our data, we assessed the consistency of sampling, as sampling effort can affect beta diversity metrics. Looking over the Beach Seine dataset, we selected 24 of the fixed sites that were consistently sampled throughout our time period. We also conducted sample-based rarefaction to standardize sampling effort across years. Before our final analysis, species counts for each sample were converted to catch per unit effort by dividing the total catch by sampling volume. Regional catch per unit efforts were also fourth root transformed to reduce the influence of the highly abundant and sporadic catches of Mississippi silversides in the dataset. Rare species with less than 10 total individuals were dropped. and individuals of the genera Lepitra and Micropterus were not resolved to species. For our data analysis, we used Bray-Curtis Multiple Site Dissimilarity Index to assess beta diversity among all regions. We used Bray-Curtis Pairwise Dissimilarity Index to compare beta diversity between regions, and we used non-metric multi-dimensional scaling to assess species correlations to beta diversity. So now getting to the results. So overall, between all regions, we've found a significant increase in multiple site Bray-Curtis Dissimilarity Index over the last 25 years in the Beach Seine survey. So basically what this means is that Beach Seine regions are getting more different in species composition over time. And if we break down the Bray-Curtis index into its components, we find that most of the dissimilarity can be attributed to balanced variation in abundance. Balanced variation in abundance is where individuals of one species are substituted by the same number of individuals of a different species. It is analogous to species turnover in presence-absence-based metrics of beta diversity. And when looking at a region-by-region comparison, we found most regions became more different to each other over time, except for Lower Sacramento versus North Delta region and the Lower Sacramento versus South Delta regions. These two comparisons can be found in the first two plots of this figure. So now we will look at species trends between the different regions over time. On the left-hand side of this figure, you will see an ordination of all sampling that occurred from 1995 to 2007, and on the right-hand side is sampling conducted from 2008 to 2019. Within this figure, all points represent the species composition of a region for a given year. The shaded areas are 95% confidence interval ellipses for each region. The vectors you see on each plot indicate the direction of increasing catch per unit effort of each species. The letter codes represent species codes used by the Delta Juvenile Fish Monitoring Program. To increase the simplicity of the plots, we only include the top 10 species most correlated to the ordination variance for each time period. So getting to the results. Overall, we saw an increased spread between regions between time periods. This increased spread between regions corroborates our findings that regions are getting more different over time. We also observed that some species were consistently associated with certain regions over time. For example, we consistently saw higher levels of carp, white crappie, and fathead minnow in the lower Sacramento River region over both time periods. We also saw consistently higher levels of three-spine stickleback, Mississippi silverside, and yellowfin goby in the West Delta region over both time periods. We also found that some species were only associated with regions under certain time periods. For example, we saw increased levels of juvenile steelhead associated with the North Delta region only during the earlier time period. And we saw increased levels of rainwater killifish associated with the West Delta region in early time periods. In later years, we saw increased levels of hardhead and Sacramento sucker associated with the lower Sacramento River region. And we also observed increased levels of tule perch and Shimofury goby associated with the West Delta region only during the late time period. So overall, what are the take-home messages of our current preliminary analysis? So our preliminary results indicate the delta has diversified over time at the regional level. We also found clear separations in species composition between regions, and these separations were increasing with time. We found that some species were consistently associated with regions, while other species varied with time period. There are a few considerations one should make when reviewing our preliminary results of this study. First, our preliminary results only include beta diversity patterns at the regional level. In our final analysis, we will also look at beta diversity patterns at site level within regions. So what this means is that we will look at, for a specific region, how different sites within that region are over time. And it is possible that we may observe biotic homogenization occurring at these smaller spatial scales. So even though regions are getting more different from each other over time, we may see that same sites within a region are getting more similar through time. It is always important when assessing beta diversity to compare beta diversity at different spatial scales to get a more complete picture of what's going on. It is also important to consider that beach seine surveys are just one piece of the ecological puzzle that is the delta. Beach seine surveys have limited accessibility and species detection efficiency within the delta. So beach seine surveys do not cover everything that's there. Current investigations using different methodologies, for example boat electrofishing, are aimed to fill in these current data gaps and increase the scope of sampling within the delta. In the future, we suggest that beta diversity analysis within the delta should include multiple gears when possible to increase the scope of our inferences and get a better picture of how beta diversity is changing throughout time within the delta. And that is all I have today for you folks. Thank you for tuning in. My name again is Ryan McKenzie and my email address is listed on the screen. And if I have time, I will take any questions. Thank you.
All right, great presentation, Ryan. And we do have some time, about 6 minutes or so for questions. Go ahead and load those into the Q&A box or raise your hand. There is one already in the queue. This is from Timothy Malinich. He asked, did you conduct a separate NMDS for each time period? Did you also combine the two time period data sets and then check for changes over time and across regions? A bigger data set would have been tougher to run, so understandable if you want to keep them separate.
Yeah, so I think with the NMDS, it's basically looking at those two it basically splits the data in half. So you have the early time period and the late time period. And the whole idea of just looking at that is just trying to identify which species make each region unique. So within the early half of the data set, how are these regions different in species composition and what species are associated with them? So that's what you're looking at. There are different ways of doing it, and I have done some of the preliminary within region stuff. So it's just trying to, NMDS is just visualizing the data. So the way that NMDS is set up is to try to visualize what the actual figures before that, just the linear regressions were showing. Yeah, so Tim, we're going to add quite a bit to the supplementary when we submit this, just to like throw out kind of like all those cool trends to look at.
All right, excellent. Any other questions?
There's a hand raised by Ted
Oh.
Summer.
Yeah, Ted, go ahead and unmute.
Hey, Ryan, great job mining the long-term data sets to answer an ecological question. You answered an obvious question, that whether there was a change in homogenization over time. I wonder if it'd be appropriate to look at the other obvious question, differences between wetter and drier years. So perhaps grouping, all the droughty years and some of the wetter years, and seeing if there are any obvious patterns there.
I think that's a great idea, Ted. I know this kind of work piggybacks off of Brian's paper, which looked at just species kind of catch per unit effort, and he did look at environmental correlations like that with the NMDS. But I think, yeah, maybe grouping them to see how does beta diversity change over wet and dry periods. That's a really good idea. I'll have to look at that. Because you do see, the one thing I will point out with that, in those figures, you do see a lot of variation in the beta diversity. Like there's a lot, it's a straight line, like, because we're looking at just overall trends up and down. But you do see from year to year, it does bounce around quite a bit. So I do think like during the drought, you do see some dips in certain areas. So I do think that grouping it dry years, wet years, like that makes a lot of sense.
Then we have a question from Tyler Pilger. He writes, is there an environmental reason for the time period split, or was this to maintain similar sample sizes?
The time periods, the NMDS again is just the visualized kind of species correlation. So I could have broke it up into, every year, but it's just trying to create a simple figure to visualize the data. So yeah, we could split it up into, however we want. I just did it in half just because, just to make it as simple as possible. But you can, it was just a subjective kind of split down the half just to see if there are major differences. And that's the thing too, we don't include every species that's, significantly correlated to NMDS, because there are more that aren't. It's just the top 10 that are shown. Yeah.
Excellent. All right, any other questions? We got about a minute and a half or 2 minutes. Not seeing anything. So I have a question, Ryan, and I may have to cut you off because it might be a long answer actually, but can you share any of the regional homogenization that you've seen? And
Yeah,
you said you've been working on some preliminary stuff.
So the one thing I have seen is within region, in the Western Delta, basically what the results shows that the Western Delta has homogenized, like those 3 sites, they have gotten more similar through time. And that's probably largely driven by Silversides in that area, just because they've gotten more similar through time, and they've kind of converged towards Silversides a lot more. Whereas the other regions have actually gotten more different sites within regions between those guys as well. And I think that's largely driven by differences in the amount of centrarchids. So some sites for some reason there's more abundant centrarchids than other sites. So there's like it's very interesting to look at that. But overall, the Western Delta region within region is homogenizing, whereas other regions are getting more different. So
interesting. Okay. Well, we are right about we're really close to 3:50. So thank you so much for your talk, Ryan, and for participating and answering those questions.
Thanks, Adam.
We'll go ahead and load up our next speaker. Is Christina Burdi with CDFW.
Hi there, everybody. My name is Christina Burdi, and I work for the California Department of Fish and Wildlife. And I'm here today to present to you on the Hunger Games, dietary overlap in larval longfin smelt, prickly sculpin, and Pacific herring in the San Francisco Estuary. Most larval fishes hatch with a yolk sac and an oil globule, and this provides nutrition for them until they start feeding. Larvae are poor swimmers, they have small mouths, and low visual acuity, and all of this impedes their ability to detect and capture prey. They are susceptible to high instances of low feeding success, which can cause delayed growth, and starvation, which can cause death. Therefore, their feeding success largely depends on prey concentration and availability at hatching. Three species that have their larval stages here in the estuary are the longfin smelt, Pacific herring, and prickly sculpin. And all of these species are native to the SFE. They all spawn in brackish or freshwater. They all have peak spawning periods during January to April. They all hatch around 5 to 9 millimeters total length. All of their larvae are pelagic and consume zooplankton, and they overlap spatially and temporally in the SFE. Longfin and herring have declined in abundance in recent decades, particularly longfin smelt. And this is partially attributed to decreases in prey availability, plus shifts in the zooplankton community overall, which have decreased their historically utilized prey, such as eurythemera and mysids. Due to all of these factors, we wondered if there was dietary overlap and therefore potential competition for food between these species. To do that, we looked at fishes that were caught January to March in 2013. January and February, they were caught by the Smelt Larva Survey, and the 20 millimeter survey in March. We compared fish that were caught at the same date and station, and we did diet comparisons by month and region. And because all three of these species did not overlap consistently in all areas, we just looked at comparisons between longfin and herring and longfin and sculpin. We examined their diets by dissecting them and counting the number of prey items in their guts. We identified the prey items to the lowest possible taxon. We calculated the diet by percent number, which typically favors smaller, more numerous prey, such as small copepods or rotifers. And we also calculated the diet by percent weight, which favors larger, heavier prey, such as mysids or amphipods. And we noted the presence or absence of a yolk sac. We also calculated the degree of dietary overlap, or the Schoner's index, which is shown by this equation here. And it looks at the sum of the differences in proportions of the same prey item in the diets of the different fish species. And we calculated this for diet by number and diet by weight, and the index ranges from 0 to 100, with 100 being a complete overlap of diet. However, greater than 60% is considered significant overlap. All right, jumping into the results here. This is a graph of the mean CPUE of all species from January to February with region on the x-axis. You can see that all species were highest in February. Longfin smelt and herring had the highest CPUE in Carquinez and Suisun Bay, and prickly sculpin had the highest CPUE in the Central Delta. When we look at the relative fish abundance, you can see that longfin overlapped with both species, with herring in the more marine areas and sculpin in the freshwater regions. All species co-occurred in Suisun Bay and Carquinez Strait in all months, and they co-occurred the most in March, ranging from Carquinez all the way to the West Delta and then up into the deep water ship channel. Now we'll look at the diet results, and I broke this up by comparisons between longfin and the two different species. First, looking at longfin and Pacific herring. Looking at the length frequencies of the fishes examined, herring was bigger than longfin overall in January and February. However, in March, longfin caught up, and they have similar sizes between both species. On the right-hand side, we have tables of the average number of prey consumed by each species in each month. And overall, herring consumed more prey, This is particularly the case in March, where herring consumed almost 3 times the number of prey as longfin. And overall, the prey number increased with increasing fish size. Looking at the percent of empty stomachs and presence of yolk sacs, 50 to 70% of fish had empty stomachs in January. Of these, 40% of longfin still had yolk sacs, and therefore still have a source of nutrition despite the fact that they had empty stomachs. There was a similar amount of empty stomachs in both longfin and herring. Yolk sacs had been absorbed by March, and only one Pacific herring had a yolk sac the entire study. One of the interesting things that we did see was we saw that longfin started feeding before the yolk sac was absorbed, and this occurred in both Carquinez and Suisun Bay in both January and February. When we look at the degree of dietary overlap, or the Schoner's index, there was significant dietary overlap in all regions and months between longfin and herring, and this was the highest in Suisun Bay and Suisun Marsh in February, where you have almost 100% overlap in Suisun Marsh during this time. When we look at their diets a little bit more closely, we have a graph of the diet by percent number on the left-hand side and diet by percent weight on the right-hand side. You increase in time as you move from the left to the right of each graph, and the top panel of the graph starts out on the more western region and moves upstream as you move down. Overall, both species were utilizing the same prey in similar amounts. Limnoithona dominates diets both by percent number and percent weight despite its small size. Other cyclopoid copepodids are also important, and Eurythemera and copepod eggs are important starting in March. And overall, there was a wide variety of prey in Napa River. So now we're going to look at longfin and prickly sculpin. Again, looking at the length frequencies of the fishes examined, there were similar sizes between longfin and sculpin in January and February. However, longfin had a wider range of lengths in March. Again, on the right-hand side, we have a table of the average number of prey consumed by each species in each month. And sculpin consumed a higher number of prey overall despite similar sizes. Looking at the percent of empty stomachs and presence of yolk sacs, greater than 85% of fish had empty stomachs in all regions in January. Of these, 30 to 60% still had yolk sacs, and again, therefore still have some nutrition despite having empty stomachs. Longfin had a high percentage of empty stomachs in February in all regions found. And there were still yolk sacs present in March, in comparison to what we saw with longfin and herring, where the yolk sacs had been completely absorbed by this time. And again, we did see fishes that were feeding before their yolk sac was completely absorbed. This occurred with both longfin and sculpin, however, only in one region each. Looking at the degree of dietary overlap, there was only one region, and that was the Central Delta, in March, with greater than 60% overlap. And note that I did have to combine the fishes in both February and March in the Deepwater Ship Channel because there was too low of a sample size in these months by themselves. Looking at their diets a little bit more closely, again, we have the diet by percent number on the graph on the left-hand side and diet by percent weight on the graph on the right-hand side. We increase in time as you move from the left to the right of each graph, and the top panel of the graph starts out in the most western region and moves upstream as you move down. Note that January isn't shown here because of the high degree of empty stomachs. Both species are utilizing the same prey types, however, they're utilizing them in different amounts. Limnoithona is not as important in these regions except for in Suisun Bay. Copepodnoplei and other cyclopoid copepodids are particularly important in February. And these fishes are eating bigger prey items than longfin and herring were, despite their smaller sizes. And this is shown by the amount of copepod adults and cladocerans, which is shown in the graph as other, that are being consumed. And calanoid copepods such as Pseudodiaptimus, Eurytemra, and Cynocalanus, that's shown here as other copepods, are particularly important in March. In conclusion, this is the first study looking at longfin diets in January and February. One of the surprising things that we saw was that Limnoithona is important for fishes during this time, and that's despite its small size and predator avoidance behavior, which is for it to remain relatively motionless in the water column. Other fishes also select against this prey. However, the good news is that Limnoithona is the most abundant copepod in the estuary, so it's good that fishes are able to utilize this prey source. All species were utilizing the same prey. However, this varied based on month and region. Sculpin and herring were eating more and bigger prey than longfin overall, and calanoids started being important in March. Another surprising thing that we saw was that some fish were feeding before their yolk sac was entirely absorbed. And this is particularly the case for longfin in the more marine regions, such as Carquinez and Suisun Bay. Looking at the dietary overlap, longfin and herring had significant overlap in all regions and months. In comparison, longfin and prickly sculpin only overlapped in one region, the Central Delta, in March. Therefore, the strongest potential for competition between all of these species is with longfin and herring in February due to their high abundances during this time, and also their high dietary overlap in Suisun Bay and Suisun Marsh. In conclusion, the potential competition for prey between these species, plus the low prey abundance in winter overall, and the decreased abundance of the historically utilized prey for longfin, such as Eurytemra and mysids, all of this could further reduce longfin and juvenile survival. And with that, I'll say that we have done a lot of additional analyses on these data to further explore the potential competition between these species, and that we are currently writing up the manuscript. However, in the meantime, you can check out our other manuscript that was just accepted on larval and juvenile longfin diets from March to June, with the title given here below. And thank you, and I will take any questions.
All right, excellent. Christina, great presentation. We do have 6.5 or 7 minutes for questions. I see Wim Kimmer has his hand up. Go ahead and unmute Wim and ask your question.
Yeah, thanks, Christina. Christina, that was good. I didn't know you were doing that. It's great. I just wanted to talk about competition a little bit because I've been really interested in this because it's kind of a fundamental ecological concept, and yet people claim to see competition when all they see is similarity of diet in planktonic organisms or planktivores. And I actually gave a talk a few years ago at a plankton conference where I asserted that there was no evidence for competition outside of fishless lakes, and nobody challenged me on it. I don't know if it's right. I was just trying to hang it out there, but you know me. I think inferring competition, particularly in the case of larval fish, is kind of iffy because they're so dilute. And I was just looking at the SLS data. The mean catch per trawl is about 50 fish. And so that means that there's about 1 fish for every 3 or 4 cubic meters, which is, how can they possibly affect the prey enough to, to influence each other is what I'm getting at.
Right, yeah, and so that's one of the things that I was particularly careful in saying that there's clear evidence of competition, because like you said, it could just mean that they have dietary overlap in this case. Obviously, we'd have to look a little bit more into prey concentration and number of fishes to truly test competition, but there is the potential for that there, considering their dietary overlap.
Well, if you do demonstrate it, you'll be the first. Actually, no. A former student of mine, Greg Jenkins, showed competition in larval herring in the Indian Ocean on the basis of slower growth inside the school. But that was the only example I could think of. It's very
well, challenge accepted, Wim.
Yeah. We've got about 4 more minutes and quite a few more questions. So thank you for asking that one. Christina, this is from Ted Summers. Christina, what was in your other diet category? Anything cool or unexpected?
I can't really say if there's anything cool or unexpected off the top of my head. I think the main thing that was unexpected for us, and probably a lot of people that know a lot about fish diets and zooplankton, is the consumption of limnothoina. I think for the prickly sculpin and longfin, the other category was mostly cladocerans.
And then Arthur Barrows wrote, I may have missed it. Were longfin selecting for L. tetraspina, or has that analysis not been done recently?
So
asked about Slater Baxter, or since Slater/Baxter.
So I haven't done that on this data set. I know that Steve and Randy did that for their 2014 paper. So I can't really say that for this data set. That is something that I'd like to explore, but haven't done it yet.
And then we have like maybe 2.5 minutes left for questions. I see Michelle Jungluth has her hand up. Michelle, go ahead and unmute and ask your question.
I am curious if you know if this was 2013, son. Was that a drought year?
I'm sorry, I can't hear you that well, Michelle.
Oh, yeah. Okay, hold on. Sure.
Maybe I need to turn up things here.
Okay, is that better?
Yeah.
Okay, great. Sorry about that. Super interesting work. Do you know if the year of that study was a drought year, just by chance? Or a drier year?
I am not sure. I'm sure that some other people that are able to answer this question.
Yeah. Well, the reason I ask, I think it was Arthur presented something earlier that suggested that the density of L. tetraspina increases in drought years in certain areas of the estuary. And in my 2017 study, which was a pretty wet year, I found very little consumption of L. tetraspina in the Suisun and San Pablo region.
Yeah, and I noticed that in reading your paper, the differences in the consumption of limno between the two studies that we've done. One of the things that I am wanting to look at before we turn in the paper or write up the manuscript fully is the abundance of the different zooplankton species during the time in these regions, to answer that a little bit more.
Yeah, great.
Okay, well, I see we have more hands up, but we are almost out of time. So I think, Christina, if you want to check out the Q&A after the session and respond to those, that'd be really great. But we're going to have to move on to the next speaker. Okay, so thank you so much, Christina, for your presentation.
Thank you.
And for answering those questions. It's really great. But we will go ahead and load up our next presentation. Our next presenter is Brock Huntsman. He's with the USGS.
Hello, my name is Huntsman, and I'm a fish biologist with USGS at the California Water Science Center. And today I'm going to talk about some work we've been doing looking at the effects of hydrodynamics on fish community structure in the Cache-Slough complex. So flow is often considered a master variable that controls fish community structure, especially in lotic systems. And the natural flow regime is often used in order to explain the evolution of life history strategies in lotic systems. And so oftentimes we think of the trilateral life history continuum, especially for fish communities when describing life history traits. And when we apply these natural flow regime concepts to this trilateral life history continuum, oftentimes we can predict what type of life history traits in fish communities that have those life history traits should be occurring depending on the natural flow regime of the system we're studying. Well, most of these concepts, specifically relating the natural flow regime to community structure and life history traits, is based on lotic systems with unidirectional flow from upstream to downstream. Well, tidal rivers provide an additional complexity to that concept by having tides influence the distribution of habitat in these tidal rivers. And so not only does flow go from upstream to downstream, but the tide influences the flow in the opposite direction. And so it's these gradients also create a complexity in habitat composition of tidal rivers that also likely influence the fish community composition as well as other aquatic organisms in tidal rivers. However, rarely are these concepts applied, such as the natural flow regime concepts with life history traits to explain fish community structure in tidal rivers. Well, in addition to just your standard tidal river, typically tidal rivers also have diverse habitat types on the riverscape. And one type of habitat are terminal channels, such as in the Cache-Slough complex or slough habitats, as well as, say, the deepwater shipping channel that have these terminal channels where there's really very little inflow, and any flow, the inflow comes from overland transport. And so in 2021, Paul Stumpner recently put out a paper showing how the landscape, specifically of these terminal channels, interact with physical processes and specifically tidal exchange in order to create very discrete exchange zones. And I'm grossly oversimplifying this, but the idea here being that as the tide propagates up into these terminal channels, those tides create very discrete habitat zones based on tidal exchange. So we have high tidal exchange zones, lower in the terminal channel where there typically is high turnover in water residence, a very low water residence time. The tidal excursion, or the distance a water particle travels upstream with the tide over a tidal cycle, creates an additional zone where all that tidal, the extent of that tidal excursion, this moderate exchange zone. And then lastly, above the tidal excursion, we have a low exchange zone upstream of it where there's very high water residence time because there's very little interaction with the tides. And so because the tides also carry water constituents with them, like such as nutrients and sediment, they also create gradients and very important habitat features that affect the community structure from microorganisms all the way up to your fish communities. And so these specific gradients caused by the hydrodynamics of these landscapes also create very important habitat template from which native fishes have likely evolved. So how do these hydrodynamic habitat features in these terminal channels influence community structure? Well, in 2016, Marine Downey put out a paper showing how the tidal excursion specifically affects the water residence time in the Cache-Slough complex. And so as you can see here, in the top 4 figures ultimately is demonstrating water residence time and how it was recorded based on hydrogen and oxygen isotopes and how that translates into water residence time. And so in the upper regions where there's very little tidal exchange, you have very high water residence time, upwards of 50 days. And below the tidal excursion in the blue, you have quick tidal turnover exchange, where residence time is from hours to a few days. And so these concepts also influence, as we had mentioned before, the water chemistry, and that also affects the productivity of the food web. So for example, what was also shown in this study is that the nutrients, specifically biologically important nutrients such as carbon, nitrogen, phosphorus, spend longer times in these upper excursion areas where there's very little water turnover or high water residence time. And because there's higher water residence time, typically that translates into greater processing of those nutrients and more biomass at the base of the food web. And so this can be represented by chlorophyll A. And that's just one mechanism of many mechanisms why the upper excursion area where there's very low water residence or very high water residence time can be important for the food web. Additionally, you see dissolved organic matter typically being higher in these upper excursion areas where there's very little exchange. And so ultimately, this all kind of translates into there's greater energy at the base of the food web that can propagate up that food web. And so this is a pretty basic concept in community ecology that shows that at the base of the food web, with more energy at the base of the food web, you typically have higher species diversity and biomass supported. And so there's no reason similar concepts don't also apply to the cash-slough complex in reference to these hydrodynamics. And so we set out to test these kind of concepts in the sense that we wanted to see how hydrodynamics shape fish community structure within terminal channels of the cash-slough complex. Our first objective was to determine or to test how hydrodynamics, specifically the zones, hydrodynamic zones above, below, or within the tidal excursion zones affect fish community structure. And then lastly, test how hydrodynamics as well as local habitat conditions affect functional fish groupings within the cash-slough complex, accounting for those life history attributes. So the way we did this is we use a stratified random sampling design. We had 3 terminal channels, the Lindsay Slough, Cash Slough, and then what we call the aggregate. The aggregate is the conglomerate of different habitat types, such as Liberty Island, which is an agricultural tract that had a breached levee. We have wetlands, such as Wildlands and Little Hollands Track. We have different channels connecting these different habitats, and then also the stair steps and the tow drain that drains the yellow bypass. So a complexity of habitat considered in the aggregate. For each of our samples, we took a sample from 2017, February 2017 to December 2018. We sampled every season with gill nets, experimental gill nets. Experimental being that they had different mesh sizes to capture small and large fishes. We deployed a gill net and took habitat measurements at every location where the gill net was deployed and measured the distance with GIS from the sample point to the closest tidal marsh. Lastly, we used Paul Stumpner's hydrodynamics model specific to the cash-slough complex in order to identify the extent of the tidal excursion. And so the stars in this figure represent locations found within the tidal excursion based on our analysis. And so upstream of the tidal excursion, within the tidal excursion, and then downstream of the tidal excursion. And so these created all the essentially zones that we use for our analysis. So for our first objective, again, looking at community structure, we use a very standard basic community analysis. We use permutational MANOVAs to look at how community structure was affected by tidal excursion zone, one of those 3 zones, channel, year, and season. And then for any significant effect from one of those discrete categories, we use similarity percentages to determine what was responsible for overall similarities. Before we did any of that, we also used species accumulation curves in order to determine if we had if we had appropriate sample size in order to test these community analyses, to use these community tests. And so the idea is with these species accumulation curves, we wanted to see, did we have a sufficient sample sizes in order to truly capture the diversity of fishes found in those habitats. And so if we reach an asymptote in the species accumulation curves, that would suggest that we did capture most of the diversity of the fishes found in that habitat. If it doesn't asymptote, then it suggests we need more samples to sufficiently sample those habitats. And what this would suggest is that 2 of our sites, specifically within the tidal excursion zone, did not reach an asymptote. And so we throw those out of our community analyses, at least for objective 1, in order to have comparable data sets. So what we found was the permutational identified channel, tidal excursion zone, and season all have significant effects on community structure. For the channel, we found that the aggregate was significantly different than the other 2 terminal channels, and this was to be expected given that the aggregate had a diversity of habitats available to it. Interestingly, the tidal excursion zones, upper versus lower tidal excursion zones, were significantly different. And what drove those significant differences was specifically a higher abundance of native fishes typically found above the tidal excursion than below it. And then seasonally, the main driver of differences seasonally was due to more hitch being captured in the fall within the cache slough complex than other seasons. For our second objective, we first needed to aggregate all of our counts into logical fish groupings based on life history traits. So we took our species matrix and used a cluster analysis in order to identify those groupings. We identified 6 groupings, the first 3 being non-native fish groupings. So the first fish grouping was a pelagic non-native fish grouping composed of striped bass, American shad, threadfin shad. The second grouping was a littoral fish grouping full of sunfishes and largemouth bass. And the last non-native fish grouping was more of a benthic grouping composed mostly of our different catfishes that were captured. The final 4 groupings were all native fish groupings, or mostly native fish groupings, with grouping 4 being Sacramento pike, minnow, and hitch, 5 being Sacramento sucker and split tail, and then 6 being tule perch. And so once we combined the counts for all these fish groupings, we used regression analysis to explore how local water quality information, distance to marsh, and distance to the tidal excursion all affected counts in each of these 6 groupings. And then lastly, use model selection in order to determine what was the most parsimonious explanation for those counts. And what we found, interestingly enough, was that all the local habitat conditions best explained the non-native fish distribution. So temperature, turbidity, depth, and chlorophyll all best explained, or were selected from model selection to best explain the non-native fish groupings. And our native fishes, surprisingly, all of them were best explained by the hydrodynamics model. So distance to the tidal excursion, specifically, native fishes were more abundant upstream of the tidal excursion, and typically were found to be more abundant closer to the marsh habitat. So conclusions, what does this suggest? Well, what we found was that, interestingly enough, that most of our native fishes, especially our native fishes, were more abundant above the tidal excursion. Now, why were they more abundant? We don't have a mechanistic explanation, or we didn't test a mechanistic explanation, but it suggests that it's likely bottom-up driven, but further exploration is needed. Interestingly, non-native fish were more abundant based or more closely affected by local water quality conditions as opposed to the tidal excursion. And so what does this mean for management? Well, so restoration and management actions may do well to consider these hydrodynamics, and how can we manipulate hydrodynamics maybe more so than cost to water in order to design any management actions or restoration actions. And why not consider this natural flow regime kind of concept in these tidal river habitats in order to design any kind of restoration actions. So a lot of people to thank, BOR, thank them for funding. Irwin was in support of this project, and obviously the aquatic ecology group, everyone there was here before me and collected all this data, and I just analyzed it. So there's a literature cited, and I'd be happy to take any questions.
All right. Thanks so much, Brock, for that presentation. we're opening this up for any questions for Brock. Not seeing anything, not yet. Okay. Kyle Griffiths, go ahead and unmute and go ahead and ask your question.
Hi, Brock. Very nice talk. I was just hoping you could say a little bit more about the idea that you got at right at the end there about increasing our restoration bang for our buck. Would you suggest, based on your results, that we just concentrate on the upper end of those back channel sloughs for restoration purposes?
So yeah, I guess I should say that I don't recommend any action or restoration action. Just provide the data as a USGS employee and the results. But I think there is there seems to be a pretty obvious relationship between mechanisms operating above that tidal excursion, whether it truly is water residence time and increased productivity up above that tidal excursion, and that productivity propagating up trophic levels. I didn't show any of the water quality information or chlorophyll A information, but a lot of those patterns are still pretty consistent with what was shown in that marine downing paper. So there's a lot of evidence that that productivity is higher. I guess what I am suggesting is considering that tidal excursion, and I'm not saying it's not considered, but I think this is definitely something that, say, John Burrow and Paul Stumpner have really tried to emphasize to me, being that they're our hydrologist, is that these, leveraging what we know about these tides could be really important in designing a lot of this restoration effort. So yeah, I guess I want to hesitate. I don't want us to give any recommendation, but considering these patterns, I think is pretty important in considering those tides.
Thanks.
Okay. So we've got 2 to 2.5 minutes for any other questions. I'm not seeing any hands. And nothing in the queue. I'll give it about 15, 20 more seconds.
Yeah, and just feel free to email me too. I mean, I'm usually pretty on, so if you have any questions. It was recently submitted, so hopefully in a couple months we'll hear something back.
All right, well, not seeing any questions. Steve Culbertson wanted to wrap things up, since this being the last session, the last presentation of the last session. So take it away, Steve.
Yeah, thanks for a wonderful session, Adam, and everybody for attending. Yeah, just a few closing words. Stephanie Fong's doing the same thing for the other concurrent session. I just wanted to thank everyone for their attendance and participation in the workshop. I've learned a lot, and I think we did a fair job pulling us all together in this virtual context. So thanks everybody for putting up with all of our arrangements this year. It'll probably be something very different next year, and I look forward to that. The recordings of all of the workshop events, those that people have agreed to have be recorded and be published, will be available on the IEP YouTube channel until June 30th. There'll be some time for us getting that up to the YouTube channel, but do look for that. So if you have some things that you've missed or you want to go back and look at again, those will be available on the YouTube channel until June 30th. You'll get an email when those videos have been posted and a link for that channel. So please watch for that. I wanted to thank Christine Job for her heroic effort in supporting us this week. This literally was not possible without Christine's input and support. So if you get a chance, please send her a little note when you see her in person. Thank her. It was largely due to her that we were able to do this. So thanks, Christine. Don't forget, take a few minutes now, maybe, to vote for the best early career posters and the People's Choice Award for the presenters. You'll see a link on the webpage to go vote for best poster, best early career poster, and best presentation. That'll be available, that portal will be available for your votes until 5 o'clock tomorrow. And then we'll let everyone know the results of that voting on Monday. So please stay tuned for that. Also, never too early to think about planning for next year. You'll see a note go out sometime early summer, midsummer. August is usually when we start our planning for the next conference or workshop. And you can always let Christine or I know that you're interested, and we'll put you on our list. But thanks to everybody for participating, and have a wonderful rest of your day and week. Thank you, Adam.
Thanks, Steve.
Thanks, everybody. We'll see you in 2023.
Well.